Okay, good afternoon and welcome. I am honored to today welcome you to the 4th annual Dr. Alan L. Bisno Memorial Infectious Disease Lectureship. We are very grateful to Alan's wife, Barbara, and son, Neil, who are here today, as well as his daughter, Susan, and the entire Bisno family for the opportunity to remember and honor Alan's contributions over the more than 30 years that he spent here at the University of Miami Miller School of Medicine and the Miami VA. I'd like to share just a few pieces of information about Dr. Bisno's history. He graduated with a bachelor's degree from Princeton and then completed his medical degree at Washington University in St. Louis. He then completed internal medicine residency at Vanderbilt. And following residency, Dr. Bisno served 3 years in the public health service, the EIS service you may have heard in the news at the CDC. What would become his lifelong work in streptococcal bacteria began during his infectious disease fellowship in 1968 with Dr. Gene Stullerman at the University of Tennessee Health Science Center. Serving as infectious disease chief for most of his almost 2 decades in Tennessee, Dr. Bisno was regarded internationally as an expert in the diagnosis, treatment, and prevention of group A strep infections. He made seminal observations regarding the epidemiology and pathogenesis of rheumatic fever and post-streptococcal glomerulonephritis, conducting a groundbreaking early clinical trial of a streptococcal vaccine, and brought attention to the importance of severe group G streptococcal infections. Dr. Bisno was a thought leader in the field of streptococcal disease, writing the chapters in Cecil's, Harrison's, and Mendel's on streptococcal disease. He co-authored national medical practice guidelines for diagnosis and treatment of streptococcal disease and rheumatic fever. And when he moved to Miami in 1987, Dr. Bisno was a professor and vice chair of the Department of Medicine at the and chief of the medical service at the Miami VA. And many of us got to know Dr. Bisno during that time. We recognized him as a brilliant clinician. Also had a very quirky sense of humor, a passion for teaching, and really notably great dedication to his patients, and never forgot the people who suffered from necrotizing fasciitis or the veterans who he loved very much who suffered from recurrent cellulitis. He effectively ensured that no ID fellow trained here in Miami would ever miss a case of necrotizing fasciitis. It has been an absolute pleasure to have the opportunity each year to reflect on Dr. Bisno's personal and professional accomplishments with his family. And I'd like to thank them again for their continuing Alan's legacy of clinical, academic, and educational excellence with this Grand Round series. So thank you very much.
Good afternoon, everyone. I'm Temur. I'm one of the Jackson Memorial Hospital internal medicine chiefs, and it's my honor today to introduce today's speaker, Dr. Madeleine Cunningham. Dr. Madeleine Cunningham is a George Lynn Cross Research Professor of Microbiology and Immunology at the University of Oklahoma Health Sciences Center. She received her PhD at the University of Tennessee Center for the Health Sciences, where she trained under Drs. Gene Stollerman and Edwin Beachy. She was a postdoctoral fellow at the Oklahoma Medical Research Foundation in Oklahoma City before joining the faculty in the College of Medicine at the University of Oklahoma Health Sciences Center after being recruited by Dr. Joseph Ferretti. Dr. Cunningham studies Group A streptococcal pathogenesis and immunity and has been supported by the National Heart, Lung, and Blood Institute, including a Career Development and Merit Award to investigate the pathogenesis of rheumatic heart disease, myocarditis, and dilated cardiomyopathy. She has a strong focus on human translational research in autoimmunity, molecular mimicry, and infection, including biomarkers of disease and outcomes. She developed human monoclonal antibody technology to unravel the autoimmune pathogenesis of human inflammatory conditions of the heart and brain, including rheumatic fever and rheumatic heart disease. Her work has also been supported by the American Heart Association and the National Institute of Mental Health. She has been a consultant to the National Academy of Sciences and represented the United States in the U.S. Indo Vaccine Action Program in India to promote research on Group A streptococcal vaccines. It's my honor today to introduce Dr. Madeleine Cunningham.
Thank you so much for that kind introduction. And also Dr. DeBlecki for the lovely presentation about Dr. Bizzo. And so when I began in Memphis, Tennessee, at the University of Tennessee, Dr. Bizzo was there in Dr. Sollerman's lab. And I was in the Department of Microbiology and Immunology at the University of Tennessee. And they thought, well, where were they going to place me to get my PhD? And so I looked at two labs, one at St. Jude and one at VA Hospital. That was Dr. Ed Beachy's laboratory. And so I chose strep over viruses. Yay. Bacteria win again. I know all of you are laughing. Anyway, but Dr. Bizzo was there in the laboratory with Gene Stollerman. And so I had a triumvirate of brilliant people around me all the time. I had Dr. Alan Bizzo, but I had my mentor, Dr. Beachy, and then Dr. Gene Stollerman, who sadly, Dr. Beachy died of lung cancer at 55, and then now Alan is gone. And Dr. Solomon died at 93, but he still came to my laboratory after all these years and helped us with a paper that we got in Nature Medicine back in 2003. I started my career in strep when I went to Ed's lab. We worked on an M protein vaccine, of course, because that was their goal. But I couldn't do that when I opened my lab. I didn't want to compete with them. So I went into rheumatic fever research. And actually, those years, they had pretty much given it up for dead, that we were never going to figure out what was happening with rheumatic fever. And so those years were the beginning of monoclonal antibodies. And so that was our first paper, was in Science in 1985, that myosin was the link between Streptococcal and HARD. And so that began all this work. And it came from Dr. Bizzo and Dr. Beachy and Dr. Solomon and all the training that I did when they were in the laboratory there in Memphis. So I certainly want you to recognize how important Alan has been in my life and also in the lives of so many people with these pictures, beautiful pictures of Barbara and Alan, and then of course his professional picture, but in loving memory of Dr. Alan Bizzo, who is a celebrated researcher and expert in group A streptococcal diseases. And always remember that he's been was author for years of the chapters in Mandel in the Principles and Practice of Infectious Diseases. And that would be, sometimes he would call me and we would talk about what was going in the chapter, that he was asking me about the sequelae. And, but of course, most of all, as Dr. DeBlecki was saying, a caring doctor, and of course, a wonderful husband and father, as well as a mentor and a wonderful example to so many. So now I have to operate this thing. If I can figure out how to move it. Let's see. Maybe I just, well, that's what I did. Maybe this will work. Oh, maybe. Oh, that's it. I'll just,
Okay.
yeah. So this is, yes, hopefully I don't do it too quickly. But of course, this is in recognition and memory of Dr. Alan Bizzo. And the title of my presentation is Autoimmunity Infection, the Group A Streptococcal Sequelae, which has been my lifelong work. And of course, at the University of Oklahoma Health Sciences Center is where my laboratory is located, in the Department of Microbiology and Immunology there for a long time, 45 years. And we've had a lot of funding over a long period of time from the National Heart, Lung, and Blood Institute for about 35 years. And the American Heart Association was my first funding in 1984. And then behind that came the Career Development Award from the National Heart, Lung, and Blood Institute. And then since then, the National Institute of Mental Health, the National Institute of Infectious Diseases, NIAID, supported our T32 program for 22 years at the University of Oklahoma that I shared with the Oklahoma Medical Research Foundation and was PI of that grant for a long time, supporting many PhD fellows and some MD PhD fellows. We've studied Lyme disease, the Brain Foundation that has given to studies of our pandas research, and then all the rest toward the end are autism and pandas with the David Judah Fund at Harvard University supporting our 1,000-patient study out of our laboratory. And then I have a financial affiliation to Exclaim. The Molecular Labs in Oklahoma City is a company that I co-founded in 2012. So of course, many of you are from infectious diseases, but many of you are just from general departments in medicine. And in fact, just met someone that knew Dr. David Kim, who is chair of endocrinology there in Oklahoma. So I know that there are a lot of you that are not in infectious diseases, but I'll try to keep this tamped down. So those of you that don't like bacteria as much as we do will enjoy listening to what we'll talk about today. But the group A streptococcal diseases are many that go from pharyngitis all the way to acute rheumatic fever. And you, I'm sure, have seen many of them. Group A streptococcal infection begins, as you know, with the sore throat or the bacteria adherence to the mucosa. And you can see that at the pharyngeal epithelium. But don't forget all the other sites that this organism could present and maybe need to be cultured in people if you can't find it in the throat. Generation of cross-reactive antibodies by B and T cells we found is important in bringing about the group A streptococcal sequelae if you don't recover from pharyngitis or the group A streptococcal infection that you may have with activated B cells and T cells producing autoantibodies and Th17 cells. I'm not sure how familiar you are with the immune T cell subsets, but Th17 is very important in protecting against extracellular bacterial pathogens and is all throughout the mucosa of the sinuses and upper respiratory tract, and also is very important in causing autoimmunity. And so it's a two-edged sword. The tissues that may be affected in rheumatic fever are many, just like in lupus, that you'll hear by the end that maybe it shares some features with systemic lupus erythematosus. But the heart, brain, joints, and skin are affected in this disease. So the major manifestations of acute rheumatic fever, polymigrating arthritis, carditis, and Sydenham-Carrier, the two we will cover today, but also erythema marginatum, a circinate rash that appears on the skin, and subcutaneous nodules are also present. And the latest revision of the Jones criteria is in circulation that was published in 2015 by Gibbons. And guidelines that I am showing here came from older versions, but generally the major manifestations are those we just mentioned, and the minor manifestations may be arthralgia and fever. With laboratory findings, elevated acute phase reactions, CRP, increased SED rate, C-reactive protein, and a prolonged PR interval. And supporting evidence, of course, for an antecedent group A streptococcal infection. And how do you do that? Well, of course, you take a throat culture or a rapid streptococcal antigen test from the throat. And also you should choose other sites like the rectum if you can't get results from the throat. Elevated or rising streptococcal antibody titers. Do you remember what those are? If I quizzed you? Anyone? Oh, yes, anti-streptolysin O. What's the other one? Anti-DNase B. So those are the two. But then After you test that and a person's positive, there's some people that are negative. 40% of the people you test will be negative. So what are you going to do? You're going to do a throat culture, or you'll run the antigen test that's so popular now, using monoclonal antibodies, once again, from the group A strep to detect the antigen in the throat. But if you get a negative rapid antigen test, you really should do a throat culture. So these are the important Jones criteria that help you identify. I'm sorry, I'm having a little bit of difficulty with it's okay, I'll get it eventually. Okay, we're going to talk about rheumatic heart disease, and we go to the global burden of rheumatic heart disease, which is primarily a third world country disease, but it doesn't mean we don't see streptococcal sequelae here in the United States, as we'll discuss toward the end of the lecture. But the rheumatic heart disease, fortunately, was only seen during the 1940s and right after penicillin was discovered by Sir Alexander Fleming. Was this treated on the wards in the 1940s, where they had many, many cases of rheumatic heart disease. So now the disease is not as prevalent. The heart disease is not as prevalent in the developed world, whereas the developing world, you can see with the large dots where rheumatic heart disease is most prevalent. And the cross-reactive immune response in the heart is very important in the development of acute rheumatic fever. We studied this using the human and mouse monoclonal antibodies to inform us about the antibodies that are present. You can see this at the top of the slide, where the antibodies are binding. They are in general to the glucanac epitope of the group A carbohydrate, and they are cross-reacting with valvular endothelium. They will bind to the strep, and they infiltrate the immune cells that react with the proteins, are getting into the tissues, and they are cross-reacting, becoming activated by proteins that are in the heart. And so as they bind to the valvular endothelium, they, of course, activate the endothelium and lead to the antibody deposition that you see in the valve, which becomes damaged. The most damage in the beginning is to the chordae tendineae, which become loose, and then the valve can't function. And before you know it, you have much scar tissue, because the chordae tendineae are very fragile. And once they become scarred, then this immediately affects the valve. Damage, remodeling with elongation and fusion of the chordae tendineae and dilation of the annular rings, valvular thickening, immobility, and collagen release, which leads to the two most prevalent antibodies. If you were to do tests on rheumatic fever patients, they would have antibodies to cardiac myosin and to collagen, which we'll talk about. Well, this is not easy to use. It's okay. I'm going to get back there. All right. So these are what were shown in the 1960s when people were investigating the antibodies in patients to the heart. And so they showed this staining immunofluorescence of the heart muscle. And so when we were investigating group A strep and we were making the first monoclonal antibodies, we asked, well, we see that, and we could see striations in some of the staining. And our antibody on the Western blot, we had the first Western blot on campus. I mean, Western blots are a dime a dozen now, but it was amazing. We could see a 200 KD band. And so we said this must be myosin, and we did prove that it was myosin, and that's why we ended up with the paper in Science, because nobody really knew what was happening at that time in rheumatic fever. I hope I'm not sure. There we go. All right. So the translation approach, as I was telling you, to the study was using human monoclonal antibodies, which, you possibly could do with some of your patients, where you use the antibodies to understand the disease, because they are a renewable resource, whereas sera from patients are not as renewable. Identification for us with the heart antigens and leading to study the pathogenesis of disease, which you can see the first monoclonal antibody, 3 that was derived from a rheumatic carditis patient in Oklahoma City, actually stains the endothelium, and it also stains the heart muscle, as they saw previously. And so this recognizes cardiac myosin in the myocardium and laminin in the valve endothelium, and cross-reacts with the streptococcal component in acetyl beta D glucosamine, which is a dominant epitope of the group A carbohydrate. And so the laminin epitope was identified that actually had similar sequence in cardiac myosin. And it's all of this cross-reactivity that helps these antibodies recognize multiple tissues and bind to the heart. Most recently, there's work that I've done in the lab that sometimes we would work on it and then we would wait a long time and begin looking again. And so recently we published this, but this study began a long time ago, where we had patients with carditis and chorea, and we decided to look at the IgG subclasses, because IgG is important. That's what is going to bind in your heart, not IgM. Because this is this secondary response that is going to target more higher affinity antibodies to the tissue and cause autoimmune disease. And so we looked at glutenac-specific IgG1, and you can see that on the left-hand side. But in B, the glutenac-specific IgG2, it's interesting. IgG2 in humans is the subclass that actually is called up to carbohydrates. And so when the group A carbohydrate response was mounted, it was IgG2. And this glutenac-specific response to IgG2 was the only one that was the highest in carditis and chorea. If you look at all the other subclasses, IgG1, 3, and 4, those do not have the same type of response that we see from the IgG2, which is significantly higher in carditis and chorea compared to pharyngitis and normal controls. And so this also was elevated in the CSF in Korea. To me, that was just phenomenal. I can't believe that we actually could see that from CSF, and it had remained preserved over time in the patient. But in the middle graph, you see the star that's on the p-value of the IgG2 subclass, because all the subclasses are listed below on the bottom. I may have a pointer. I don't know if I do. No, I don't think that's one. It's okay. I think it'll be all right. Look at just the middle graph and you can see the star at the top, and that's IgG2, and it's specific to glutenac. And that is in the spinal fluid of people with Sydenham chorea. And we just recently finished this study. We started it many, many years ago and finally decided we had to finish it because the IgG2 is the primary dominant subclass that binds to the heart valve. And so it's likely it is the group A carbohydrate epitope, the glutenac epitope binding to the valve. You can see a little IgG3, and actually recently Nicole Moreland, Dr. Moreland in Australia, just identified that IgG3 in general was higher in patients with rheumatic heart disease. And then the IgG2 binds greatly, and this is the glutenac-specific epitope, and you can see that there's very little binding in the others. And if we look at another set of hearts where we look at 4 other patients, you can see that if you compare with all the other subclasses, really the one that stands out is IgG2. And this was why we decided we had to publish this, because we looked at it on the slide. You didn't even need a microscope. The IgG2 was just red right on the edge of the valve. And so it was, to us, amazing that the link between the group A Streptococcus and the heart had come down to the glutenac epitope. And this, of course, deposits, and when it does, and you start out the process of rheumatic heart disease, you get VCAM1, which is a adhesive molecule for T cells. And so it will draw the T cells to the surface of the valve, and that's stained in red, the VCAM1, which is upregulated on rheumatic hearts. And then you can see the CD4 T cells having adhered, and they are extravasating through the valve into what is an Aschoff body. So those of you that have never seen an Aschoff body, that is one there in the valve tissue. You can see that. It's a granuloma. That's what an Aschoff body is. And you know, if you're all practicing and you know what a granuloma is. So that the immune attack in rheumatic carditis is to, of course, to the group A Streptococcus, but then the B cells and T cells produce antibodies and cytokines that bind to the valvular endothelial cells, making the valve vulnerable to T cell extravasation with T cells that are to the M protein. Because T cells don't recognize carbohydrates, and the M protein itself, actually the antibodies can bind to it, but some of the antibodies can bind both of them, can bind to the M protein and to the T cells and to cardiac myosin. And if it's the brain, it's another alpha helical protein. It's tubulin, is the internal biomarker there for those antibodies. So when T cells get into the heart, whatever they see must activate them in order for them to cause damage. And so we know that these T cell clones from rheumatic heart disease patients, they react with M protein. But what about the other proteins? And you can see myosin is the most reactive one, and that's why it's the important molecule in the disease. The other two that we see cross-reactivity with the antibodies, tropomyosin and laminin, are further they're not as reactive. You can see that they are further, the concentration is less. And so helping us to understand how the T cells help the B cells make the antibodies, because that's why we get the anti-myosin antibodies in rheumatic fever, because they cross-react with the strep, not because it's myosin in the myocardium. A lot of cardiologists get really bent out of shape about why is it myosin? Myosin's not in the valve. Well, you also have collagen antibodies, and that comes from the collagen that's expressed in the valve. But overall, the T cell response in rheumatic heart disease, which is the other reason we decided we had to publish this, is because the Th17s are just tantamount. They are all in your sinuses, and when you get any kind of upper respiratory tract infection, they're going to respond. But specifically to these extracellular bacterial pathogens, Th17 is a new T cell subset compared to the others, Th1 and 2, that were discovered many years ago. But now we know the Th17 is the cause of many autoimmune diseases. And so the strep activates it, and it's repeated streptococcal infections that cause this, one after another. And at our lunch, we're going to have a discussion about treatment, because the question is, what do you do when a child comes in and you don't know, and you can't get a throat culture? And what do you do? I want to know what you're going to do. I don't know the real answer either. But if you let it go, I know that I have this whole handful of patients now that call me every day that they're sick because they weren't treated. And I don't know what the answer is, but I know we don't want drug resistance, but we also don't want all these people that are crazy out there because of strep. So we'll talk about that further in a moment. But you can see on this slide, I don't have the pointer, but you can see the middle is gamma interferon, and you're going, well, that's not Th17. Correct, it's not. That's T helper 1. But the Th17s become T helper 1s over time, and they make a lot of gamma interferon, and that is how you fight group A strep, is with this response. But the Th17 are first. They're what are seen and are activated. They open the blood-brain barrier so that antibodies can get into the brain, or they migrate cells, go to the heart from the tonsils, because the tonsils are full of Th17 cells, and that's where they're getting to the heart from, is from the tonsils. So So then the multi-step pathogenesis, just to go over this, and we're going to leave the heart behind and go on to the brain. But the outer valve endothelium is so important. This is where you break the integrity of the valve with the autoantibodies, ironically, to cardiac myosin. But they bind to laminin on the endothelium. And the endothelium becomes inflamed. You see the VCAM1 activated, then the step 2, the T cells extravasate. Just follow me through the slide. Gamma interferon, Th17 cells are not listed on here, but you just saw them. You've got predisposition, just like in lupus or any other autoimmune disease with HLA. It's different in different nationalities. Like in Turkey, it's one HLA. In Italy, it's something else based on people's nationality. Eventually, there is breakdown of the internal valve, and as you get scar tissue in the valve, what do you get? You get more blood vessels. So every single time you have another strep infection, you cause the infection to explode in the valve, and it just becomes worse and worse, and that's what happened, and why children lost their valves during the 1940s and 1950s, because disease exploded in their valve and they could not survive. And so now we're very lucky. We saw, in a way, surgery has solved rheumatic heart disease, and also penicillin and the antibiotics here in the United States and Europe. But it's not solved elsewhere. And that's why, of course, it's important to consider a vaccine, which I won't really talk about today. But to conclude, the glucanac-specific IgG2 that I showed you, defined in our study as the dominant IgG subclass in both rheumatic heart disease and in the CSF and Sydenham chorea blood, and which adds to our basic understanding of the sequelae. And it could be that it distinguishes and separates pharyngitis from acute rheumatic fever, and it could be the risk marker. In other words, we might be able to identify these children before they ever get rheumatic fever, and that Th17 and Th1 responses are very important, and reminding you that Th17 cells are the dominant T cell response against extracellular bacteria. So this is a long list, but let me go through them very quickly. These are our current knowledge on the biomarkers: cardiac myosin and collagen autoantibodies. And CD8 positive collagen reactive T cells are recently found in valves of women. 90% of the valve cases are in women. Remember lupus? All the 90% of the cases are in women. Guess what triggered those CD8 positive T cells? Estrogen. Made them release perforin and granzymes. It made them active, and it made them cause chronic valve disease in these women. So TGF-beta overexpression is not proven, but it's a hypothesis. Transvalvular pressure gradients are a potential. HLA haplotype, you know that that's important in autoimmunity. Other therapies can be considered, hydroxychloroquine, and that's published in Circulation in the recent years. And maybe even consider rituximab if necessary. And streptococcal vaccines, of course, we need to know what the antigen is that leads to the cross-reactive antibody. Is it in protein, or is it the group A carbohydrate? It's still up in the air. So at that point, we've covered rheumatic heart disease. We'll go on to the brain, which is Sydenham chorea, and another disease that you may like or you may think I'm crazy, but it's pediatric autoimmune neuropsychiatric disorder associated with streptococcal infections, or PANDAS, and they're not little animals running around. They may be little animals running around in their brains, but anyway. Some of you may have not seen Sydenham chorea, so I want to show you that. Let me try, because it showed earlier, and I wanted to try to get this to work.
The play button's right there.
Let's see. Well, oh, okay, let me Oh,
Okay, right here.
Is 12 years old.
12 years old.
Yeah, put your hands out in front of you.
Put your hands out in front of you. Just for those of you that's never seen a case of Sydenham chorea, I thought it would be interesting for you to see this.
Once more.
Once more, and she's trying to touch her nose. And so since I have this, I'll take the moment. I'm going to talk about Sydenham chorea for quite a while, but don't worry, I'm not going to keep you forever. Is that in pandas, it's not that. It does not look like that. But you can have some chorea with pandas, and they are small choreiform movements of your fingers and toes. I'm sorry, I didn't bring a video of that, but very important sometimes to identify that. And lots of arguing whether pandas is Sydenham chorea or it's not Sydenham chorea, or does it even exist? So I get it, all of you may have a different opinion of it, but this is a lab where we found out what it was. And all I can do is show you the evidence, and it's pretty convincing. It's just, if you don't read, you can't know it. Yeah, I'm good. So these movement disorders, first, of course, is Sydenham chorea, which you saw, and then PANDAS, which is obsessive compulsive behaviors and hyperactivity and emotional ability and tic-like symptoms. I'll try to be fast. I really don't have a clue what time it is, but I'll move as fast as I can. So Sydenham chorea and PANDAS have the same staining of the caudate putamen. You can see that, the CSF. Whereas the control CSF is negative from various other types of diseases like yeast. Again, we took the same approach to this translational approach. We found out that in Sydenham chorea, this was the paper that Dr. Solomon, I think, nudged and helped me get into Nature Medicine in 2003. And the first thing we identified was that these antibodies weren't Guillain-Barré syndrome. They reacted with a different set of lysoganglioside and mono and disiloganglioside antigens. And you can see the main antigen in red, lysoganglioside. And this is generally important. It's seen in our hands in Lyme disease, in the neuro-Lyme, and in Lyme disease. In chorea, you can see that the lysoganglioside in red, those are 3 patients. These are from NIMH, and they're in red, reacting with the lysoganglioside. And then in convalescence, the antibodies are reduced. And they're cross-reactive. They react with the membranes of neuronal cells. And you can see a commercial anti-ganglioside antibody looking just like the Sydenham chorea antibody on the surface of human neuronal cells. And these antibodies, actually what we discovered is they signal neuronal cells. They don't kill them, they signal them. And that's where the movement is coming from. And so if we look, Sydenham chorea, this is in our first study of PANDAS and Sydenham chorea of the patients. We see that chorea is much higher than PANDAS. And then non-PANDAS group is normals, ADHD, some other things. So we took the human monoclonal antibody we made in Sydenham chorea. I don't have time to show you all the slides of it, but the most important is that we made beautiful human monoclonal antibodies from Sydenham chorea, and they reacted with these things that I told you, the lysoganglioside and tubulin, and we put them into a transgenic animal. And the basal ganglia, where they entered. They were produced by the B cells, so they're in the blood, and we broke the blood-brain barrier, and they got in the brain, and they went straight to the basal ganglia, and they were in the ventral tegmental area and the substantia nigra. And this is published, and I wish it were a higher journal. It's in the Journal of Immunology. Of course, they'll come slit my throat for saying they're not high enough, because They, anyway, I won't say anymore. And you can see that there are few in the cortex, but there are none in the hippocampus at all. And the hippocampus is where lupus, neurologic lupus is involved, and the NMDA receptors. Here, these are the dopamine receptors. And so we then ask the question, is there evidence for dopamine receptors in this disease? Antibodies. And so sure enough, of course, we looked at the monoclonal antibodies and patient sera. You see the patient sera lined up. They all signaled because it's a negative receptor where it in the inhibitory receptor for those that are neurologists or brain people. You can see that they go down compared to the untreated. And then if we look further and look at the monoclonal antibody, you can see what dopamine does and that the antibodies mimic dopamine. They behave like dopamine and actually signal the D2 receptor on neuronal cells, and then the D2R, the transfected D2R cells are signaled, and that's the bottom graph. So it shows the dose response. So they're animal models. I'm not going to spend a lot of time on this slide, but they're mice that the antibodies deposit in the basal ganglia. There are Lewis rat model of rheumatic heart disease that also worked, and the animals couldn't hold a food pellet that if we immunized them with strep. They couldn't hold a food pellet, and they went cuckoo when you sprayed them with water. They just never stopped grooming. So they had a lot of behavioral changes. You can transfer this with IgG and do it intrathecally or by blood. And then the transgenic mice you just saw. And the antibodies, that's about D2. There's also D1. And so D1 is important in pandas. And I'll finish the lecture by just showing you why I believe that. But it's interesting with D1. D1 actually becomes more sensitized when the antibody is on it. It becomes more reactive to the dopamine. So you can imagine a child or a person having both of these antibodies very high, because when we see patients that have very high D1 and D2, they're psychotic. And so this has been published in the position paper in Lancet that says that D2, they didn't list D1 yet because it's pandas, but they listed D2 as being important in one of the different psychoses because they have all the different ones, the NMDA receptor, now there's the dopamine receptor. So just wanting you to realize this because it's something that people have resisted because they just want to say they're crazy, but they're not just crazy. They have encephalitis. They have autoimmune encephalitis, just like lupus. People have an encephalitis if they have lupus psychosis. So when the D2 receptor is signaled, what happens is too much dopamine is produced. So you can imagine if you have both of them, you've got too much dopamine produced. It's sitting on the D1. It's making it more sensitive. And so then it's easy to see how a person could not be well. This is just our animal model where the animals did not hold food. You can see GAS, group A strep, in immunization. They dropped the food pellet. They couldn't walk on a beam, and they had crazy grooming when you sprayed them with water. And they didn't have activity deficit disorder. So we looked at a separate group of Sydenham chorea patients from Israel. And so these patients also had D1 and D2. They mimicked the same thing that we did with the NIH group of patients with Sydenham chorea. This is Dr. Hilla Ben-Pazi, my wonderful collaborator for many, 20 years from Israel. And then the dopamine receptor antibody ratio, nearly beat her to death to get this data. I said, I won't publish it till we correlate it. And so it's the D2 titer over the D1 receptor titer that correlates with the symptoms, which is irritability and movement. So then we took three different cohorts. We have cohort 1, which was the first pandas cohort, the first 50 patients discovered by Dr. Sweeto. The second cohort is also from NIMH, but it was a little different. It did not have the choreiform movements in any of the patients. They were just straight pandas. And then my own large 1,000-patient group that we only have 850 samples we can use. But we took these and first looked at chorea, because that's something surely I keep thinking everyone believes that there is chorea in people. And so there's no predictive value of D1. These are ROC curves. You may not be familiar with them. I didn't used to know them until people talked to me about diagnostic antibodies. And so these are the area under the curve. You can see on the right that the D2 has great predictive value for Sydenham chorea. And then the anti-D1 receptor is a biomarker for pandas, but not D2R, even though they may have some D2 and they may have the little movements. The D1 shows up in cohort 1, 2, and 3 to have predictive value for pandas, but not for Sydenham chorea. And so just a few details, and I'll be finished, is that the novel human monoclonal antibody that we first got in pandas was amazing, and it reacted very strongly with peptides in the extracellular loop of the dopamine receptor. I'm not showing you actually the peptide work, but I'm just showing you the loops. The peptides were actually on the plate in an ELISA. And so you can see certain peptides, EL1A, and that's borne out for the other two monoclonals that we've also prepared from pandas, and that it reacted in the top very right-hand corner. Oh, good. I've now got a little marker. You can see the red staining, and this is D1. That's the only one that has the D1 in it. The rest are controls. And the signaling below, I'm just going to move on. And these are human sera. These are not monoclonal antibodies, because I knew you would want to see the patients that we had studied. And this was just recently published, November 8th, in the Journal of Clinical Investigation Insight, where you can see the controls SC in pandas for the D1 mediated receptor signaling of the pandas patients. And the reason it's not so different, although it's significant, is because they're GPCR receptors, and you have to have radioactivity or really hot signaling in order to pick them up. And so we're just happy to be able to report significance from them and have tests, because most tests you can't see the difference. And so then the bind and activate D1R, just to drive that home, that's just expansion. And these are two new monoclonal antibodies that we prepared, and they do the same thing as the other one that we prepared from patients. We'd like for them to do the same thing as the human sera, because you don't want to show a minor incident coming from the blood. And so the diagram shows that with the neurons, you can see the pandas antibody binding to D1. That's what we propose, that D1 is primarily for pandas and D2 is primarily for Sydenham chorea. Whether you want to believe they're both Sydenham chorea and one is lesser and one is more for the movements, but the way the terminology came out of the National Institutes of Health was that pandas was going to be called pandas, and it was separate from Sydenham chorea because it was OCD and tics. Whereas in chorea, you might see some of that, but only minor. It will be more movement. You will see, like the video that I showed you. Whereas in pandas, if they have choreiform movements, they're very small, and you really have to look for them and have the child hold their hands out. And so this had just came from our Nature Reviews paper with Dr. Jonathan Karapetis and Andrew Beaton, showing the neurons and that there are other antigens besides the dopamine receptors, lysoganglioside, which seems to be more prevalent, the antigen in Lyme disease when you get these, because you can have other components, other infectious agents causing pandas. And the question is strep the beginning of all of it? You know, we may not know. We may not ever know, because it may be that in a patient, they get strep, it's not diagnosed, and then they get Lyme disease, and they were already triggered with strep before. I don't know the answer to it either. What it's called is PANS, Pediatric Acute Onset Neuropsychiatric Syndrome. And this syndrome can be caused not only by strep, but by Lyme disease is one, with Borrelia burgdorferi, the flu, influenza, mycoplasma. These are some staph. I've seen staph abscesses that would lead to this in the sinuses. And the autoantibodies we know activate CAMK2 because they're activating neuronal cells in the basal ganglia and releasing too much dopamine, which then sensitizes the neurons to dopamine and makes everything so magnified that patients may become psychotic. And there are numbers of patients that, they were trying to kill people or kill themselves. And so it's something that it's really detrimental not to recognize this disease in people, because they're not to be locked away. They're meant to be given what? What would you give an autoimmune encephalitis person to treat them? IVIG or plasma exchange, because the plasma exchange, of course, works very well on all these people. Even Sydenham, Korea, works very well. This has been proven at NIH already. But you're not going to want to go to that kind of trouble to do it unless you've got a facility that automatically runs plasma exchange all the time. So what you'll want to do is use IVIG to try to get your patient well. If you need to. Sometimes it's not, the PANDAS is not so great. You know, it's about what can you tolerate as parents? What can you tolerate as an individual? Whether you go for IVIG or whether you take long-term antibiotics. Remember, that's what they do with rheumatic fever. You know, people forget because we don't have so much rheumatic fever here, but we've got this whole country filled with these children that are having strep, and they're calling me every day. And, you know, I don't even know some days how I can get any research done, because I'm like you're on the phone with your patients all day. And I think, oh, what am I going to do? So, because I can't turn them away, it's just their lives are just totally upside down with these neuropsychiatric diseases in their child. Their whole family, the only child they can focus on is that one, because if they don't, they're just going to be totally lost. And getting them treated is insane because no one wants to treat them. No one wants to treat them. So it becomes difficult to find people. They come to me because they need care and they don't know what to do. So the disease symptoms are a two-step process. One is what we've told you about the autoantibodies, but the other is you got to open the blood-brain barrier. Maybe you have the antibodies and your blood-brain barrier never opens and you're not ever sick. But you have to, this has been shown, and IL-17A is the component that breaks the blood-brain barrier. That paper was published not too long ago in PNAS, explaining how most likely they enter the brain. And the first studies on Th17 and strep were done by Dr. Patrick Cleary, who is a group A streptococcal researcher that is now retired, but he's still alive and kicking, and did great work. And then Dr. Agaloo was the neuroscientist that worked with him to show about the brain. So there are a lot of people to thank, because it's been a lot of years, and my laboratory and all these people, particularly Suzanne Roberts, who is an MD that decided she wanted to do research, and she did all the work on the valves. all that VCAM work. She did all of that in 1 year. And we got a paper in the Journal of Infectious Diseases with her work. And Dr. Galvin, who created monoclonal antibody 3B6, that he did all the studies on the heart. Dr. Curvan, who did a lot of the PANDAS work, she's on 4 of the main papers. And then actually Dr. Menendez is hoping to take my place in PANDAS and eventually be a faculty member. And she's a finishing postdoc now and hopes to become assistant professor very soon. And then all of our collaborators from Vince Fischetti, George Veazey, who's a cardiologist in Utah, the people in Hawaii, India, Brazil, Israel, Dr. Ben Pazzi, Dr. Sweeto, and then just they're listed here again. And then my lab group, this is Dr. Curvan, and then Dr. Mertens did all the T-cell clones. We would have never known without her. And then finally, just our funding again to thank Dr. Michael Janicki. He graduated from OU Medical School and recently died of pancreatic cancer. And he's the one that gave us the $100,000 from the psychiatry department at Harvard to do the 1,000-patient study. We wouldn't we wouldn't know about the antibodies, the group, without that. And so thanks to my lab, Dr. Menendez, who's really working so hard now, is hoping to become your PANDAS expert very soon.
Thank you, Dr. Cunningham, for sharing that elegant work with us and really an illustrious career in this particular area, showing us how tenacity and science can actually make breakthroughs to important diseases. I'm sure there are lots of questions. We don't have a lot of time. We're at the top of the hour, but maybe I can just ask a quick question. Given the fact that these antibodies are the sequelae of the infection, which then result in the sequelae of these cardiac and neurologic manifestations. When we think about vaccines, and given the current chief of our Department of HHS, are we concerned at all that any vaccines that promote antibody responses could elicit these types of antibodies, especially as you develop vaccines for this? Is there any concern about that, inducing these types of neurologic and cardiac effects?
Okay, well, I'm going to give the first answer. We all took the COVID vaccine after it was tested for 3 months or whatever, a short period of time. So that's the first answer. You can take your own idea with you from that answer. The other answer is that we've been working on a group A streptococcal vaccine since I worked on my PhD. That's 50 years ago. We still don't have one. I'm still working on it because we recently, one of the big pushes is now for the group A carbohydrate. And what is really interesting is that the epitope that I use for the vaccine, not for the vaccine, for the cross-reactivity, is the N-acetyl beta-D-glucosamine epitope hooked to BSA, bovine serum albumin, and that coats it to an isoplate. The antibodies from animals that are given the group A carbohydrate, the whole thing, I don't see much cross-reactivity. I'm getting ready to report that at the Lancefield meeting in Australia. And these are companies that are, providing me material because they want to try to move forward with the group A carbohydrate vaccine. I can tell you that John Zabriskie tested the group A carbohydrate. It doesn't have the avidity and power that M protein does, but nothing will, because the M protein is 200 serotypes, and that's highly specific, and it's a protein. The other is a carbohydrate, but it will protect against all group A strep. And so to me, if it will work and we can show that it will be safe, I think it's really important.
Great. Thank you very much. I want to again extend my thanks to the Bisno family, whose generosity has made this lecture possible. We look forward to seeing you next year at the Bisno lecture in good health. And I remind everybody to check the chat for the code for the CME and MOC credits for the residents as they exit. They can see the QR code. And I wish everyone a great day and look forward to seeing you all next week. Thank you.